Hi everyone, I'm Madam Tava here. This week lesson is the continuation of the previous lesson. So we are still in chapter 1 enzymes. So this week you're going to learn about inhibition, reversible and irreversible, competitive and non-competitive, allosteric regulation, naming and classification of enzymes. So at the end of this lesson, you should be able to describe the basic principles of enzyme inhibition, describe the allosteric regulation of enzyme, define the classes of enzyme. So before we learn about the inhibition, allosteric regulation, and the classes of enzymes, let me do a small recap about the previous lesson. So in previous lesson, you learn about the factors that affecting the enzyme catalytic rate of reaction. There are a total of 6 factors. However, you learn about 5 factors in previous lesson, such as the pH, temperature, enzyme concentration, substrate concentration, and cofactor. For pH and temperature, all the enzymes have their narrow range of pH and also the optimal temperature. So if above the optimal pH and temperature, the enzyme will be denatured. Therefore, the rate of reaction drops. For enzyme concentration, the rate of reaction increases as the enzyme concentration increases. For substrate concentration also, the rate of reaction increases until the saturated point. Above the saturated point, the rate of reaction will be constant. Cofactor means the non-protein molecule that needed by the enzyme to work more effectively and efficiently. Okay, let me show the example of the reaction of the cofactor. The shape of the active site of apo enzyme is not same as the shape of the substrate. So once the cofactor binds to the active site of apo enzyme, it's known as halo enzyme. It means the cofactor together with apo enzyme. So now the shape of the active site is complementary to the shape of the substrate. So the substrate can bind to the active site. So this is how the cofactor helps the enzyme to work more effectively and efficiently. So we begin today's lesson. So the first subtopic is inhibition. The inhibitors that cause the inhibition. There are 2 types of inhibition, such as irreversible and reversible inhibition. Irreversible inhibition is a type of permanent inhibition. However, the reversible inhibition is type of temporary inhibition. There are 2 types of reversible inhibition, such as competitive and non-competitive inhibition. Okay, based on the competitive inhibition, the competitive inhibitor bind at the active site and prevent the substrate binding. So it means the competitive inhibitor compete with the substrate for the active site. But this situation can be overcome if there is a high concentration of the substrate. So if there is a high concentration substrate, the competitive inhibitor release from the active site, then the substrate can bind at the active site. So the example of the competitive inhibitor is malonic acid. Based on the non-competitive inhibition, the non-competitive inhibitor or the allosteric inhibitor, their target is not the active site. The non-competitive inhibitor will bind at the site other than the active site. It means allosteric site. So once the non-competitive inhibitor bind at the allosteric site, it will make changes on the shape of the enzyme, and also the shape of the active site also change. So the substrate cannot bind at the active site. Once the non-competitive inhibitor release the enzyme back to their original shape, and then the substrate can bind at the active site. So this situation also can be overcome by increase the substrate concentration. So if the concentration of the substrate is high, the allosteric inhibitor release from the enzyme, and then the enzyme reverse back to the original shape. Then the substrate can bind at the active site. So the example of the non-competitive inhibitor is amino acid isoleucine. So amino acid isoleucine non-competitively inhibits the enzyme threonine deaminase. Okay, next we look at the irreversible inhibition. Irreversible inhibition is a permanent inhibition. It means the inhibitor binds permanently with the enzyme by covalent bond, and it will affect permanently the enzyme. Then the enzyme will become denatured, and then it lost its catalytic properties. For example, the cyanide inhibit the enzyme cytochrome oxidase, which found in the electron transport chain that used in the respiration. So this inhibition caused by non-competitive irreversible inhibition and then can cause death. So the examples of the inhibitors such as insecticide, the cyanide and arsenic. So basically the irreversible inhibition permanently affect the enzyme compared than the reversible inhibition. So this is the example of animation of the competitive and non-competitive inhibition. So based on the animation, you can see here, if let's say there are high concentration of the inhibitor, so the substrate cannot bind at the active site. Okay, now the substrate concentration high, so the substrate can bind at the active site. So if let's say high concentration of the inhibitor prevent the substrate and they compete with the substrate for the active site. In non-competitive inhibition, when you see here, there is a high concentration of substrate. So the substrate can bind at the active site. If let's say the inhibitor bind at the allosteric site, it will prevent the substrate binding. So once the inhibitor release, the enzyme reverse back to the original shape. Thus, the substrate can bind at the active site. Okay, next is allosteric regulation. Allosteric regulation means is a regulatory mechanism in which the non-substrate molecule binds to the site other than the active site of an enzyme, and then it will induce the enzyme to change its shape. So the non-substrate molecule means allosteric regulators such as allosteric activator and allosteric inhibitors. So the allosteric regulators, they bind to the allosteric site of allosteric enzyme, and then it will stimulate the enzyme to change its shape. Allosteric enzyme means they made up of more than 2 polypeptide units, and each of the units, they have their own allosteric site. So you can see from the picture here, the allosteric enzyme, they made up of 4 subunits. So each of the subunit also have its own active site. And then the allosteric enzyme can change between the active form and also the inactive form. Allosteric inhibitor involved in allosteric inhibition. However, allosteric activator involved in allosteric activation. So first, the allosteric inhibitor binds to the allosteric site, and then it causes the changes in the shape of the enzyme, and also the shape of the active site also change, so that the substrate cannot bind at the active site. Next, the allosteric activation. So once the allosteric activator binds to the allosteric site, what happened? The shape of the enzyme change compared than their original shape, and then the active site also change. So now the substrate can bind to the active site. Allosteric regulators such as the allosteric activators and also the allosteric inhibitor. So the binding of the allosteric activator to the allosteric site of the allosteric enzyme will stabilize the active form of the allosteric enzyme, and then the inhibitor. So once the allosteric inhibitor binds to the allosteric site, it will stabilize the inactive form of the allosteric enzyme. Allosteric inhibition. As I mentioned earlier, allosteric inhibitor binds to the allosteric site and then it will change the shape of the enzyme and also the active site, and it will prevent the substrate binding. Feedback inhibition is a metabolic pathway in which the end product controlling their own rate of production. So the end product will bind to the first enzyme and then it will prevent the substrate binding. So this is the example of the allosteric feedback inhibition or end product inhibition. End product inhibition is a form of the inhibition mechanism. So the allosteric feedback inhibition involves more than one enzyme. And then the end product acts as a non-competitive reversible inhibitor which binds to the allosteric site and then it will prevent the substrate binding. So the initial substrate is threonine. Before the end product binds to the allosteric site, the initial substrate threonine can bind to the active site of the threonine deaminase and then they will undergo the series of the reaction until they produce the end product isoleucine. So here the isoleucine is the one act as a allosteric inhibitor which bind to the allosteric site and then it will prevent the substrate binding. So generally the end product inhibition is to reduce the wastage of the products by the cells inside the body. If let's say the concentration of the isoleucine is less inside the body, so what will happen? The isoleucine will release from the allosteric site and then the enzyme threonine deaminase will be reversed back to their original shape so that the substrate threonine can bind to the active site and then they will undergo the series of the reaction and then they can produce the end product isoleucine. So the end product in this allosteric feedback inhibition turn off its own production. The next is naming of enzyme and the classification of enzyme. The enzyme named based on the two parts, substrate that they act on and also the type of the reaction that they catalyze. So the second part is actually ends with the suffix ASE or ACE. For example, enzyme lipase. So the first part represents the substrate that the enzyme act on. So the substrate in lipase is lipid. So there are 6 classes of the enzymes. So this classification is based on the classification system that introduced in 1961 by IUB, International Union of Biochemistry. So there are 6 classes of the enzymes such as oxidoreductase, transferase, hydrolase, isomerase, lyase, and ligase. So to remember easily all these the classes, you can use the acronyms using the first letter of each of the classes. So based on this, I form the acronym over the hills. So O represents the class OV. Next is naming of enzyme and classification of enzyme. The enzyme names consists of two parts. The first part is the substrate that they act on, and the second part is the type of reaction that they catalyze. So the second part normally ends with the suffix ase or ase. For example, lipase enzyme. So the first part is substrate. So in the lipase enzyme, the substrate is lipids. Enzymes can be classified by the chemical reaction catalyzed. There are 6 classes of the enzyme. This classification system is introduced in 1961 by IUB, International Union of Biochemistry. So there are 6 types of the classes such as oxidoreductase, transferase, hydrolase, isomerase, lyase, and ligase. So to remember these classes, you can use the acronym over the hills. O represents the oxidoreductase, T represents the transferase, H hydrolase, I isomerase, L lyase, and L ligase. So to recall back all these classes, you can use the acronym over the hills. So the first class is oxidoreductase. Based on this class, there are 2 types of the reaction involved, such as oxidation and reduction process. Transfer of hydrogen and oxygen atoms or electrons from one substance to another substance. So the examples of the enzyme such as cytochrome oxidase, lactate dehydrogenase. So the name of the dehydrogenase represents the reaction of the reduction. And then the oxidation represents the reaction involves oxidation reaction. The second class is transferase. So based on this class, transfer of functional group from one substance to another substance. The functional groups, for examples, the methyl, acetyl, amino or phosphate group. So the examples of the enzymes are transaminase kinase, alanine deaminase, acetate kinase. The third class is hydrolases. In hydrolases, there is formation of 2 products from a substance by hydrolysis process with addition of water. So the examples of enzymes are lipase, amylase, and sucrase. Lyase means non-hydrolytic addition or removal of groups from substrates. The examples of the enzymes are decarboxylase, aldolase, isocitrate lyase. Isomerase, intramolecular rearrangement. It means the rearrangement of the atoms within a molecule. So the examples of the enzymes are glucose phosphate isomerase. Then the ligase. Ligase means joining of 2 molecules with breakdown of ATP. So the examples of the enzymes are acetyl-CoA synthetase, phosphokinase, DNA ligase. Actually, the hydrolases and also the lyase, they involve formation of 2 products from one complex substrates. However, in hydrolases involve the addition of the water molecule, but in lyase does not use the water molecule. So that's all for today's lesson. Thank you.